INAD stands for Infantile Neuroaxonal Dystrophy. It's a rare disease, and it's caused by pathogenic changes in your DNA. When you have two copies that are carrying this, you basically lead to a protein that is either broken and it can't do its job, or it's missing, and so it can't do its job. This protein is important for maintaining the membrane of your cells, amongst other jobs. What we're trying to do is add back that protein, and one of the ways we do that is with a therapeutic called gene therapy. Here at the Rare Disease Translational Center at the JAX laboratory, we're doing an experiment where we have a mouse model that has the same manifestation that patients do, so they just don't have enough protein to actually function. And then we're testing gene therapy to add back that missing protein and see if it's safe, which we can then use to move forward to apply for a clinical trial in people. I think we're really one of the first steps in getting a therapy into people, right? Like, you have to start somewhere, and mouse models are a great way to do that because humans and mice share so much biology, and we have the tools to manipulate the genetics in order to make the mice mimic human patients. And so with that powerful tool in hand, we can really start to understand, again, if therapies are safe and if they're working. And that's something that we have to have in order to move forward to the FDA or other regulatory bodies. I think we were lucky because one of the mouse models that we are using is actually a very good mimic of the human disease. So we had a really good platform to start our therapeutic testing because we understood what was happening in the mouse and how well it translated to people. So we were able to actually move pretty quickly in terms of these studies, which doesn't always happen. I think a successful outcome is really what we're seeing right now, which is we're seeing dose-dependent extensions of survival in our mice. So our mice that are treated with low dose are living longer. Our mice that are treated with a high dose are living even longer. And we're seeing this at a statistically significant level, which means, these are actually measurable changes that we can detect. I think support from patient-driven foundations for rare disease research, like the support from INADCure, really provides funding where there might not be funding anywhere else for this type of research. And for rare disease, you really have to start somewhere. And so the funding that's provided by the foundation and its supporters is really the first step in getting research like this started and going. And that's what drives progress, and that's what drives us to a clinical trial. I mean, Lena's a fierce driver of change, right? She's really pushing to get things going where they need to go, her and her team really. And I don't know that INAD would be in the same place without her and her foundation moving things forward the way she has. Collaboration is huge in academic science, and particularly for rare disease, because we don't have time to not work together. And so collaborating has been really important for this project and all of the projects that I work on. I think the moments of hope are, the days we get back pieces of data and we start to see differences between, our treated mice and our untreated mice for the first time. And when you hear about other rare diseases where things are moving into clinical trials or a new drug gets approved by the FDA, you can see the light at the end of the tunnel, and I think that's very motivating as well.